Good morning. Thank you for joining us for this presentation in the Toxicology and Societies speaker series. I'm Ruth Sofield. I'm an environmental professor. I'm an environmental toxicology and chemistry professor at Western Washington University, and I'm joined by my co-host, Tracy Collier, who's a toxicologist retired from NOAA's Northwest Fisheries Science Center, and he continues his work as a consultant. Tracy, if you could turn on your camera and say hello.
I did. I'm here.
Hi, Tracy.
Hello, Ruth.
Good to see you.
Okay.
We would like to begin by acknowledging that we at Western Washington University are on the ancestral homelands of the Coast Salish peoples who have lived in the Salish Sea Basin throughout the San Juan Islands and the North Cascades watershed from time immemorial. Please join us in expressing our deepest respect and gratitude for our Indigenous neighbors, the Lummi Nation and Nooksack tribe, for their enduring care and protection of our shared lands and waterways. And now I'd like to go over a few logistics. We will save questions for the end of the presentation, but you can use the Q&A feature on your screen at any time. You will be muted throughout the presentation, so we will use this Q&A feature to interact. You should have different viewing options on Zoom, and we suggest that you use speaker view so that you can see our speaker and the presentation at the same time. The focus of our speaker series is to provide understandable examples of how events with a toxicological consequence or component have consequences to people and societies. We are happy to welcome Dr. Markus Hecker today. Markus is a professor and Canada Research Chair in Predictive Aquatic Toxicology with 24 years of experience in conducting research in ecotoxicology and chemical hazard assessment. He's considered a global expert in environmental risk assessment, Ecotoxchogenomics, hazard characterization of contaminants in native fishes and amphibians, and development of alternatives to live animal testing. Today, Markus will talk about next generation tools to prioritize chemicals and for environmental management, the Ecotoxchip. If I were to put this into my own words, and Markus can correct me, this is a tool that can be used to provide information using the genes of organisms to tell us what a chemical is doing to them. And Markus works with several managers, government and other types to figure out how to use this information to make decisions. And so we're really happy and lucky to have Markus here. And so I just wanted to welcome you, Markus, and I look forward to hearing what you're going to present.
Well, thank you very much, Ruth. And again, thanks to you and Tracy for inviting me to speak to you today. Before I start, so I will try to share my screen, and I hope I do that right. Okay, can you see my screen?
Not yet.
Okay, share. Does that work?
We can see it. Thank you.
Okay, perfect. So again, thanks for inviting me to this exciting speaker series. And well, just going back, this actually started as a discussion between Ruth, Tracy, and I at a beach in Greece, where Ruth asked me about what we think about doing something beyond just the science piece. And here I am now admitting or agreeing to give a presentation at this series. So I'm not sure what I got myself into, but I hope I will do your expectations justice. The topic I'll be speaking about today is not so much an environmental issue as a development of a tool that helps us to address those issues we have with exposure to environmental chemicals. And before I start, oh no, my thing doesn't work. Okay, sorry. Before I start, I of course like to acknowledge my co-authors, not only on this presentation, but my co-investigators on this project. This is a large-scale effort that is supported by Genome Canada. And we have three co-principal investigators, Nil Basu on the top left, Doug Crump right, to the right of Nil, and then myself, and 5 other science leads that help us manage this project. And if you want more information, I only have 40 minutes today, so there's lots of information we can share through this project. You can find this on the following website, and I'm assuming this presentation will be made available to people as well. So there's some links in there. If you want to learn a bit more, please follow those and send me any emails if you have questions or like further information. So to start, I think we're all aware that we're facing a real issue with chemical contamination in the environment. And just to put this into perspective, we had a brief discussion earlier today about the Flint, Michigan lead issue with drinking water. But just on a global perspective, you may like to know that chemical contamination now is one of the largest causes of premature deaths in the world. So there has been a pretty recent report in The Lancet that stated that we have about 9 million people dying every year from exposure to environmental contaminants. And this is, in a perspective, 3 times more than AIDS, tuberculosis, and malaria combined, or 15 times more than any armed conflict, violent conflict, including wars we have on this planet right now. So this is a real significant issue we are dealing with. No. So to deal with this issue, and there's a chemistry that society is using right now that makes our lives better as well, there have been large-scale efforts across the globe that try to manage that and try to make sure that the chemistry we're using, the chemicals we use in our everyday products, in pharmaceuticals, agricultural products, are safe for us to use, and also for the environment. Here's 3 of the main programs globally that currently regulating the use and registration of chemicals. The European Union's REACH program, you probably all have heard about, of course, the US EPA, and then in Canada, where my home turf is, it's Chemicals Management Plan or program. So one of the major issues we're facing right now, and regulatory agencies are facing, is the sheer amount of chemicals that have to be regulated. This estimated society is using approximately 350,000 chemicals, of which about 100,000 require regulation. To put it again into perspective, we have data, really reliable data, on about 10,000 of those chemicals yet. So that's less than 10% of what we need to regulate, which becomes a huge issue because those chemicals are already in use. On top of this, we will have regulations that require us to test environmental samples, ranging from industrial effluents, for example, municipal effluents, water, sediments. And in Canada alone, this is estimated to cost about $6 billion a year just for pollution control and prevention measurements. And then on top of that, we have the continuous development of novel or new chemicals. It's estimated that about 500 to 1,000 chemicals enter the market every year. Tens of thousands of chemicals are assessed every year for their purpose to be entering the market. So there's a huge amount of testing requirements here as well. So if you look at the status quo of testing right now, so how do we do regulatory chemical testing, or for R&D purposes, we do that using live animal tests. And this is becoming a very big issue because it costs a lot of money. It takes a long time, and it's ethically questionable. So here's some estimates I pulled from the US EPA website. They estimate that each chemical takes about $1 to $20 million 4 years and about 1,000 animals or more to develop for one compound. Or if you look at the European REACH regulations, they're mandated to test about 101,000 chemicals over the past decade. And the estimates here came into about 9.5 billion euros, which is about 13 to 14 billion US dollars, and about 55 million animals. So it becomes apparent very quickly that this is not really a feasible way to go about assessing all those chemicals. So if you think about, there's 90,000 chemicals out there we need to still assess. If we use the classic approach, we will be decades before we have any reliable data on these chemicals. So there has been movements now to go into alternative ways of assessing those chemicals that are much more rapid, much more reliable, but also use less animals and are cheaper. And here's again our classic paradigm of testing chemicals. So we have a contaminant like dioxin on the left up here. We expose, let's say you are a human toxicologist or a mammalian toxicologist, expose your rats to this chemical, and then you see if it's reproducing, if it's dying, growing, or developing normally, or maybe developing cancer. So these are the classic ways how we have assessed those chemicals in the past. And then we regulate. Well, if we understand why or how those chemicals interact with organisms, maybe that helps us not to have to do those long-term apical outcome or pathology types of tests anymore. And this has been proposed by a landmark report by the Natural Research Council of the US that was published in 2007 that really pushed the regulatory and the toxicology community towards using alternative ways of making those assessments, using mechanism-based information. If we understand how a chemical interacts with an organism, maybe we can come up with predictive models that help us better understanding why and how chemicals cause effects, but also predict which chemicals would be the high priority ones here. So it's similar to you going to the doctor, getting a blood sample taken, and then they look at some biomarkers, maybe your LDH is very high, you have a high blood sugar, or certain enzymes are out of whack. Well, they will make an inference from that and see if there's potential health risks you're facing. So it's a similar idea to this type of approach. And this really has accumulated in the late 2010s or 2010 into a new framework that was established, which we term the Adverse Outcome Pathway Framework. So this framework now that has been adopted by many scientists, but also has been pushed by the US EPA and the European Union quite extensively, in Canada, is a framework that tries to identify pathways of toxicity, meaning that they try to identify how chemicals interact with molecules very early on, with a receptor, for example, with a protein, with a membrane, with a nerve maybe, and then how this progresses through a series of events to an apical outcome that we currently regulate our chemicals on. And again, this would be at the organism response, where you see if animals die, if they reproduce, or if they have, for example, impaired reproduction. So the idea here really is to link those effects together. It's like a domino effect, if you wish. So you flip over the first domino, and then it results in a cascade of events. But the idea is, if we understand this very left part here, how chemicals interact with those biomolecules, or the immediate molecular responses that trigger, which happens very fast. This is a response that happens within hours or a few days. Then we can make predictions probably to effects that we care about at the right side, where we regulate on right now. Again, here's the analogy to a domino effect. So if we really understand this very first force that has to flip over this very first domino, then we can probably infer what happens further down the line, where serious effects on health may occur without having to measure those effects. So why now? Why not 30, 40, 50 years ago, when we were aware about all those issues that we were facing with environmental contamination? Well, one reason for that is that we have had a huge evolution in technologies over the last decade or 2 decades. And one example I like to use in this context is the first human genome sequencing project. I think most of you are aware that this didn't even happen that long ago. About 17 years ago, we had the first human genome fully sequenced. And this project took a long time. It took over 10 years, up to 15 years, and it cost about $3.6 billion US. So again, put this into perspective, this happened less than 20 years ago. I think they finalized 15, 17 years ago. Today, we can do the same job in several days to weeks for less than $5,000. And that's because of a machine that you see at the bottom right here, a benchtop sequencer. I have 2 of those in my own laboratory. It's very easy to use, to run. You don't have to be an expert anymore. And you can literally run a whole human genome or any other genome in a couple of days or a couple of weeks, depending on how much you know about it already. So there's been a huge jump in technology that is now available to everybody that we didn't have even 10 or 15 years ago. And this really has sparked the dawn of what we call new approach methods. So again, coming from the classic way of, we call it kill them or count them in toxicity testing. You throw animals in a tank or in a cage and then expose them and then see if they die or reproduce. Now we have a whole plethora of ideas of how we can improve that by using mechanistic assays, by using in vitro animal alternatives, or even in silico predictive systems like computational modelings, for example. We have now approaches that look at a structure of a chemical and then they make a prediction whether it's toxic or not. So all those things have really evolved very, very rapidly over the past 20 years. However, while we have a host of those approaches and methodologies being published in the scientific literature, lots of scientists have had great ideas, that have developed some tools, we still face a lot of opposition to the implementation of those approaches. And most of this has to do with lack of trust by the regulatory community. We have been regulating chemicals for decades, and regulators are often not scientists. Regulators are often people that make decisions. They manage data, and then they look at the policies behind them and make a decision. And they're very comfortable with the classic way of, well, the animal is dying, well, we have a problem, of course. If you give them a gene is changing up or down, well, they're scratching their head and say, well, how much does it really tell me about a true problem we have? So there's this lack of trust, this lack of knowledge at the regulatory side. That's one of the main barriers to the uptake of those new approach methods, we call them. Also, we don't have standardized approaches and validated approaches yet. So this has been what I call the Wild West of toxicogenomics. So everybody went off. We all have now our own sequencers. We can run proteomes. We can run fancy machineries, but it is all individual scientists that do this type of research. And we all, as scientists, are famous for having great ideas and then run with it by ourselves. So it's usually not a concerted effort where you bring the regulators in, where you bring industry in, where you bring all those partners in that ultimately going to be using those tools. So these are some of the main barriers yet that we have to overcome. And this is where the project I'll be talking about today is coming in. It's called the Ecotoxchip, or a next generation toxicogenomics tool for chemical prioritization and management. And really what we try to do with this project is to develop, standardize, and then validate and commercialize such a tool, such a new approach methods tool. And do that not only for us as scientists or for the scientific community, but do that in junction, in combination, in partnership with the regulators and with industry and societal partners in this endeavor. So, learning a bit more about this project, here's again our traditional way of how we have done toxicity testing. So we have a toxicant symbolized by their skull and bones over here. Then we expose live animals, fish, birds, mammals, rats, for example, or monkeys. And then we get some data on survival or reproduction, and then we make a decision. Well, what we try to develop through this project is an approach that doesn't use live animals anymore. It uses embryos that haven't hatched yet in eggs. So this is for 3 species: fish, amphibian, and birds, and we expose them while they're still in the egg and for a very short period of time, only 4 days. Then after 4 days, we extract genetic material from those animals. Again, under legislations currently, these are not considered live animals until they self-feed, independently feed. We extract genetic material from those animals, and then we look at the genetic material and see if the chemicals we have exposed them to have shifted the balance, the homeostasis of their molecular scenario status. And then this is fed into a cloud-based evaluation platform termed Ecotox Explorer. That's the second product we developed through this approach. It's a cloud-based online molecular data evaluation platform. You feed your data in, and then it gives you information whether you have certain pathways, certain biological processes that are affected in a manner that would help or that would inform potential adverse outcomes on effects we usually regulate right now. For example, development. Is there a cancer risk in there? Is there genotoxicity? Is there reproductive toxicity? So why do we think this is feasible? Why do we think this will be successful? Well, there has been examples of similar approaches in the human health arena that are now used for exactly the same purpose that we developed this ecotoxicity tool for. You all may have heard about cancer panels, gene panels for cancer screening, or there's human liver toxicity screening panels as well that are commonly used in the world of human toxicity assessments. Also, we have developed a pilot product. It was a reduced avian tox chip, we called that had a much smaller number of biological endpoints on it, but it has been taken up by the regulators. They're using that for environmental screening right now. In Canada, they use it for bird screening. Many ocean or coastal birds that live in high-risk areas, for example, those that are polluted with oil. They have been successfully adapting those and using those, and they really have pushed us to develop those tools further into a more general toxicity testing screen. The project, again, is aligned along that adverse outcome pathway that we have talked about a couple of minutes ago. We see that same chart here on the top. I've simplified that at the bottom. The concept is pretty much the same. It starts with a molecular initiating event, so where a chemical and a biological molecule interact, and then it triggers this domino effect to cause those individual effects or population types of effects that we care about. So what we are developing is a molecular screening tool at that very front part of that pathway. So at the gene regulation level, that has 384 different endpoints embedded on a chip. And here you see a representation of such a chip. Each of those little gray dots in there is a different biological endpoint that's in a microplate. The plate is about that big overall, and you can run it in a Qpcr machine, as we term it. And we develop those chips for 6 current species that are of regulatory relevance, 2 fish species, 2 bird species, and 2 amphibian species that are the key indicators for ecotoxicity testing. Then what we establish was when we test those chips with the birds, we establish what we call a toxicity fingerprint. So that gives you an indication of what's wrong with the organism, what biological processes are disrupted in this organism. And then this is fed into that data evaluation platform I talked about a minute ago. And then this helps you making a decision of what the biological activity and what the potential risks are. And in parallel, we'll be developing a guidance document for regulatory agencies, for industry and other societal components to inform them about how we can use that tool for this purpose of chemical safety and environmental risk assessment. The project is aligned along 4 tiers or 4 prongs. And as I said before, there is a lot of pushback right now for those to use those new approach methods for the reasons I just listed. So before we even started into designing an actual product, we went with something called GELS, and I will explain in a second what that means, where we looked into societal needs. So that's a whole group of social scientists that went out and just tried to understand what the current regulatory landscape is, what the needs are, what the doubts are, and how can we design a tool that really meets regulatory needs at this point. Then with that information, we used an approach called design thinking to design the actual tools we wanted to develop with the chips and the data evaluation platform. And then, of course, there's no tool development without validation. So the third stage then was a very thorough validation of the tools we developed together with our partners. And then finally, and that's the process we're moving in pretty soon, is, well, once you have validated your product enough or your tools enough, you want to have it adopted, ideally through the OECD, that you're going to have a test guideline that now allows you to use that type of approach in regulatory testing. So let's talk about GELS first. So this is a unique approach or requirement by Genome Canada, who's the funder of this project. It's a national genome institution that funds large-scale projects like this one. And GELS in this context stands for Genomics and its ethical, environmental, economic, legal, and social aspects. And what the purpose here is really to make sure that the project you're doing is feeding or is fulfilling societal needs. That GELS team, or the GELS team we have worked with together, has done several sub-projects. First thing is just understanding the landscape of toxicogenomics. What's out there? How many people use those types of tools? Have they been adapted? Maybe there's countries, there's legislations where those tools already are adopted. Who can run those types of assays? So here's a global map. The different colored dots are institutions that have either developed similar tools or that have capacity to run those or have trust in those tools. We have also developed a timeline. Again, toxicogenomics is a rather new field of research. However, there's, you believe it or not, a 3,000 or 5,000 year history in doing this type of assessment. So this is all captured here, and it really helped us understanding the evolution of the field, not only of environmental toxicology, but also then those new approach methods, what has worked in the past, what hasn't worked in the past, what tools have found trust by regulators, and that helps us informing them to do the tool development right on our end. Here's a summary of the outcome. So one of the main purposes was, what are the current societal needs? And then from the societal needs, what are the barriers, the hindrances we are facing? And this is captured in this graph. I don't go in detail through that. It's part of that publication I showed earlier, and this is openly available if you want to listen or want to learn a bit more about it. But in a nutshell, the green boxes here are the drivers for each of those aspects, and then the yellow, the red ones are the barriers, so that we need to still overcome. And this then has been moved into a process. So all this information has been collected and gathered and moved into a process we call design thinking. So we took information from the needs of regulators, of industry, of other societal partners that are involved in chemical safety assessment, environmental risk assessments, and try to distill down, okay, what's really needed here? What do the users need? And then together with them in a partnership, we have defined what we need to do to kind of overcome those barriers and then come up with the idea to develop those chips and the platforms. Right now, we are in the process where the prototypes we have developed are tested by the user community, and I will talk about this in a minute. So we pretty much have undergone the entire process. But what we learned through the process was that it really is critical that you engage your user community, that you engage regulators to really inform those products if they ever are to take those up. Then using this information we have gained here, we have moved into designing the actual chips and the online evaluation platform. And again, here's how this platform looks like. It's this chip, which is about 15 by 10 centimeters large. It has 384 little indentations in there, and each of them has a different biological response element attached to it. Many people may ask, why did we use Qpcr arrays, microplates? From a scientific perspective, this is old news. So we have been using those technologies for over 20 years by now. And there's much more fancier equipment, like I showed you early on, sequencers, for example, that you can put in your lab, and you look at whole transcriptomes, all the genes that are regulated at a time in an organism. These are tens of thousands of genes. However, what we learned through the initial social science approach and questionnaires where we asked regulatory agencies and other users is that most of them are not very comfortable with those new technologies. However, everybody has used a Qpcr machine, at least in their testing labs and their regulatory labs. So we really base that on a known and trusted technology. And there's companies out there that produce those already. I showed you there's tools in the human health arena where they have liver or cancer panels. So it's the same technology that we choose for that. So the idea was not to develop a new technology, but rather a new approach. And then the next part is, what are the right species to develop those for? Again, there's hundreds of thousands of species we want to protect out there. Which ones are the right ones? And of course, we started with the low-hanging fruit. So there's a few species that we use in regulatory testing in the laboratory. In North America, that would be the fat-head minnow for fish, Xenopus laevis, or African claw frog for amphibians, and the Japanese quail for birds. And then we also wanted to have more environmentally relevant species. For example, African claw frogs is not a native species in North America, and neither is the Japanese quail. So we expanded that tool set then to 3 species that we regulate on based on environmental monitoring programs, the rainbow trout for fish. This is a wood frog. We switched to leopard frogs by now for amphibians, and then double-crested cormorants for birds. And as I said, we wanted to get away from live animal testing. Again, there's a lot of ethical considerations about using large and vast amounts of adult live animals. So we try to move this all into an embryo-based system where we only expose very early life stages that are not considered live animals under current legislations. Then how did we go about picking the right endpoints? We wanted to make sure whatever the tools look like at the end, it's relevant to predicting true biological hazards or impacts or health effects. And we used a 3-pronged approach again. First one is the classic, what we call the Toxicologist Approach. So we did our in-house experiments with a diversity of chemicals where we tried to identify molecular fingerprints that were predictive of apical or health outcomes of regulatory relevance. But in parallel, we knew that there is a host of information already out there. There's databases over databases that have gathered toxicogenomic information from any type of organism, from rats to mice. There's some human data on there. There's a lot of fish data on there and amphibian data. So we mined those databases to extract biological responses that always kind of tend to be the main effectors in those testing approaches. And then finally, we went to the regulatory agencies, we went to industries, we went to the regulators to ask them, okay, what do you need? What do you regulate on? What are you comfortable with? So we used that 3-pronged approach to inform the design of those chips. So the first part, again, was this in-house doing experiments with 8 very diverse chemicals, anything from a metal to a pH, a petrohydrocarbon, to hormones that we care about right now, E2, for example, or trenbolone, softeners, pesticides, you name it. And the way we went about this is we had aligned our exposures along current OECD test guidelines. So these are test guidelines, how toxicity tests are currently done under regulatory requirements. And we exposed those animals using those same test guidelines. But then in parallel, we did a very short-term test for only 4 days for most of the species, where we took samples for our molecular analysis, and then we continued them out as per standard requirements. And the idea here was to test and see if we can take a very short-term exposure, both in embryos and adults, to calibrate our embryo assays, to make a prediction for an apical outcome. Then we assembled all this information, again, along that concept of this adverse outcome pathway I had showed earlier, to extract information on molecular fingerprints. So again, this was the idea, how can we extract the right information on the molecular level that helps us make predictions on those apical effects later on. And from here, we call that our molecular fingerprint that is predictive of those apical effects. This is just to show you how many exposures were done. That we did a total of 52 exposure studies. These were all dose response based exposure studies with 6 different species and 8 different chemicals. So there's a very rich data set available to draw from. Then we used this information. As I said, we had a 3-pronged approach. We used our own in-house toxicity data, about 100 to 160 genes that we put on those, or endpoints we put on those plates, are from those experiments we did in-house. And then we have another third of the genes based on what the regulators gave us they would like to regulate on, or they comfortable regulating on. And then we used a third of those bioinformatics extracted genes from all those databases. So now we have built our chips. We had those very first, we call them version 0.1 chips in hand. So now we wanted to see how well do they perform. Do they truly do what we promised our partners that they would do? So we went through a very extensive validation effort to make sure that they do that. And there's 2 main pieces to this validation. One is what we call a technical validation, that just demonstrates how reproducible are those tools we produced. So the technical performance pretty much was assessed here. And then we had what we call a biological validation, or we call it fit for purpose. Again, the ultimate goal of those tools is to predict health effects that we can regulate on, that we can make a very, very safe decision, but also a confident decision that what we see is really translatable into an impact on, let's say, normal development, reproduction, and so on. And here's just some data, some examples of how that panned out. The first thing we assessed was how reproducible are those tests. And what you see on the left is a graph that shows you the coefficients of variation. So this is just a measurement of variability that you have after you run the same thing a couple of times, the same plate a couple of times, or the same samples a couple of times. The lower that number on the y-axis, the better the test performs. So that means in this case, we have 3 laboratories, 3 of the key laboratories that developed the test. University of Saskatchewan, Environment Canada, and McGill University, and all of our variabilities were less than 5%. The average actually was about 2%, 1.9%, which is very, very good. So that means we are very confident in the data technically that we can generate here. The next step then is we compared the data we generated with those chips as a new technology with some of the large-scale sequencing data we had generated in parallel, just to see if the platform really produces the same data than some of the larger-scale high-throughput platforms like RNA sequencing. And what you see here is the very good congruence between the data we have generated with the chips and the data that those large-scale sequencing platforms give you. So that's a linear regression, and if they fall all along the same line, that means we have a really good predictive power of the chip for other types of endpoints. So we have great concordance here. And one of the most important parts, of course, is then how good is the test actually to predict effects on biology we care about. I give you one example here for a fish study we did with ethinyl estradiol. This is a component of birth control pills that we worry about because it's released into streams and rivers and lakes through municipal wastewater effluents. There's a lot of data available on those as well. So we did studies that looked at the gene expression, protein expression, histology, and actually reproductive endpoints for those chemicals. And again, we aligned this data along this adverse outcome pathway that follows us through this presentation. And here's what we found. So this is representative, a simple depiction of those adverse outcome pathways. On the left side, you see your molecular response patterns. So you see the estrogen receptor organisms, the chemical binds to the receptor. Activation of estrogen signaling is what we found through all the sequencing data and the chip data we have generated. And then we could link those to the red boxes on the right, which are your apical outcomes. These are your health effects that you want to predict. And we could very clearly show that in embryos, at least, we had significant impairment of growth, of normal development. So those fish that were exposed to low concentrations didn't develop gonads. They probably would not be able to reproduce when they're adults. We had renal function failure. So they're very severe effects. So we were very good in making those predictions. Then the other part, I talked about this already a little bit, that we developed is a data evaluation platform. Again, if you develop a tool like this, it's not meant to be used by experts all the time. So this is really supposed to be used by regulators that often don't have bioinformatics background, that don't know anything about toxicogenomics, how to make use of all that data. So the idea was to build a very user-friendly interface here that's cloud-based, it's free, you can upload your data very easily with one push of a button to help you interpreting the data. But it also has versatile functions for expert users that want to explore by themselves. So they have multiple ways. So you have a regulatory function, non-experienced user function, you have a highly experienced user function there as well. And the purpose of this is, from a regulatory perspective, to give you something we call like a traffic light output. So you put your data in, it analyzes what biological processes have been disrupted, have been altered, and then it tells you at the very end, red, you have a problem, Houston, we need to regulate. Green means, okay, this chemical doesn't seem to perturb anything that we deem of relevance or importance. Or orange, that means we need to probably produce more data. We have an uncertainty here. The features this program has, or this platform has, are many. So you have visual analytics, you can run your own statistics. It even prints a report at the very end. It has a textual reporting function where you can analyze your data and it prints it out and it gives you a description of what you just have found. Another key component of this tool is what we call fast BMD. That's a benchmark dose software that you can use for genetic or toxicogenomic data. And one of the main reasons for this is that most risk assessments are done based on toxicity thresholds. So it means based on the Paracelsus paradigm, everything is toxic at one concentration. Even sodium chloride is toxic if you eat too much of it, it kills you. However, you need a lot of it, or sugar. So really what our job as toxicologists is to find out what are the problematic compounds that act at very low concentrations. And so far we have done that by dose response modeling for apical outcomes. So we thought maybe we can do something very similar for transcriptomics data. That we can look at a dose threshold, a concentration threshold based on the responses we get, and then predict an apical outcome. And this has been already done for some data sets. So there on the left, you see a human data set that's been done with molecular data that was linked to cancer data. And you see the data really nicely aligns along a line. So there's a very strong linear regression between the toxicity thresholds that you measured with molecular tools versus the one that you measured with your classic cancer assays, for example. And then the right shows a very similar picture for ecologically relevant species. I think there's some fish data in there, and there's some other mammalian and bird data in there. We applied the same to our own data sets. So here's one example. This I had showed before, the adverse outcome pathway for ethinyl estradiol. So we plugged all of our transcriptomics data into this tool, and then we asked it to give us toxicity threshold values, which are on the x-axis of the left big graph here. This is the concentration of the chemical needed to cause a certain type of level of effect. And then we looked at how many genes do have to be dysregulated before we can distinguish a certain effect. And this is the thresholds we calculated that were somewhere between 0.2 and 6 nanograms per liter. And then we compared that, which you see on the BMD with a little asterisk in here, a biological relevant endpoint that has been produced for the same species before for reproduction. So at what concentrations does this chemical cause an effect of failure on reproduction? And you see it's exactly the same data you generated with the toxicity for genotox, for transcriptomics data screening and your apical outcome where you have reproduction. So that gives us really confidence that the tools we're developing are really predictive of something that we care about and that's important from a regulatory standpoint. And this is pretty much all the data I will be showing. I'll just be wrapping up in a minute or two here. So at this point in this project, we have developed most of those Ecotox chips already. We are in the process of validating them. They're actually available already for 2 species, for Japanese quail and the fathead minnow. They have shown to really be very, very powerful predictive tools. We're going into large testing labs right now that do test those for us as well, and some regulatory agencies to see how they handle with them and how happy they are with those approaches. And of course, this is a huge effort that is not only a small group of scientists that started, though there is a huge cooperation behind all of that you have seen. So on the left, you see the research and development proportion of this project. There's the 3 key institutions: Environment and Climate Change Canada, McGill University and University of Saskatchewan, but then we have a group of government partners and private industry partners that helped us develop the tools. And right now we're going into the adoption phase where the OECD is a member, part of US Army Shell. We have Bayer right now interested in that as well, and the European Chemical Authority as well. And with that, I conclude my presentation. Here is just a picture of the group that has made this all possible. This is our annual project meeting somewhere in northern Quebec 2 years ago when we still could meet in person. So it's truly a large group effort to make this all work. And with this, I'll be quiet. I would like to thank you for dialing in and for hopefully your interest in what we have done here. And of course, I'm happy to take any questions. And as a message, if you have any questions I would like to discuss further, or have any materials, my email is down here. And I'm thinking that Ruth will be happy to share that, or Tracy as well. Let me know if you have any questions. I'm happy to connect with you then.
That's great. Yeah, thank you, Markus. We do have a small number of questions right now coming in through Q&A, and that number could continue to increase. Really neat to see what is toxicology in the 21st century. And I think we've made enormous improvements since when I was a grad school student. And so I'm really thankful that you were able to come and show us what's happening, kind of like what is cutting edge work. We have at least registered, I don't know how many people have actually come into this speaker series today, but we have people who range from very little scientific background to, I can tell you, we've got practitioners, we've got consultants on this call. We have a couple of postdocs from Switzerland who they actually are working on the gene chip with brown trout and would like to collaborate with you. So we have the range of backgrounds, and so I just wanted to ask you what is kind of a basic question. I think if we could go back to the Ecotoxchip and think about what those little spots actually are, and if I kind of set this up, it's really neat that what you're doing is you're looking at taking a whole organism, so taking a fish and seeing what a chemical will do to that fish, and then trying to relate that to what's happening on the Ecotoxchip. But what is actually on the Ecotoxchip? Is it a gene? And how do you pick the gene? How do you get it on there? If you could do that kind of in some plain speak language for us, that would be great for us to understand. And then how do you do you take a chemical and add it to the chip, or how do you like what does that process look like?
Okay, well, let's start at the very beginning. So the chips have been designed as I just explained. So there's been lots of experimentation going in there. There's databases out there that have looked at what genes are dysregulated, what genes are up or down regulated, or that then are indicative of something that we care about, like a health impact that we currently measure, like cancer, for example, or like reproductive failure, you name it. So this was the core of this. So what we have done, we have extracted those genes through our own experiments or through other databases that are out there, all by talking to people like regulators or industry or other partners that we have worked with. And then we took this assembly of 384 genes that are now on the chip. So the chip is literally like a microplate with a whole bunch of little holes in there, and each hole has a tracer for a specific gene. So these genes are picked by the pathways of biology that they inform. So each biological process starts with the expression of a gene, and then that triggers a biological response. It's made into a protein. This protein then is what I call a machine or an engine in our body to do something, breaking down food, for example, kind of transporting certain things into a cell or out of a cell, you name it. So there's a lot of our normal biological functioning represents or is depending on that. So we picked those genes that are, so each of those wells is a certain specific gene that informs those processes. Well, you can always do a gene, it's easy to pick, but the tricky part now came in, how do we pick the right genes that really help us understanding what's changing biology that we care about? Well, having cancer, you all care about. That's a big problem we have. Having diabetes, well, this is a pretty significant issue for somebody. For a fish not to reproduce, well, you're doomed. You don't have any babies, your population goes down. So these are really the key things we care about. So the tricky part was to really make this link, to make sure we pick the right genes, which are in each of those wells is a different gene, that help us inform if they pushed, like this domino effect, if they're pushed in the right direction or wrong direction, what does it mean biologically that we care about? Does that answer your question?
I think that was great. And I think that might answer some other questions that we had in here. Okay, so there's two more questions that are kind of related, and these relate to other organisms. So what do you think about, I mean, the way the questions are phrased is extending this to phytoplankton, invertebrates, marine mussels, organisms that are sessile in that specific question. I personally am interested in marine organisms and thinking about how what you're doing could either translate into and how we could use it to interpret what's happening in other organisms, or whether at some point other organisms would have a gene chip, an Ecotox chip developed.
Yeah, very good question. And again, the question we get very frequently. So just to start out, this is a start. So we wanted to kind of have a harmonious way of doing something, and we had to pick a couple of organisms that we could work with. Again, there's a plethora of organisms out there we like to do that for. That's why we picked the organisms we did, because they are currently used in regulatory testing. But we are aware that these are very limited. They're only a very small number of, like fishes alone. If you just even take the invertebrates out, if you just take fish, there's about 30,000 species of fish on this planet. That's more diverse than if you compare birds to mammals. So how do the two fish species we picked are really predictive of all of that? I don't know, and they're unlikely to be. So we had to start somewhere. However, the neat thing here is we translating now the chips we have built to other species. So there's a lot of interest. Zebrafish came up. Most people in the human health world use zebrafish or not the fat minnow or a trout. So we're doing that. We're having a definite chip we'll be building hopefully pretty soon. Right now, it's a funding thing. So we're having an application in because lots of people do invertebrate testing or algae testing. So EPA has right now a challenge, for example, that they want to have those animal groups involved. So yeah, those chip developments are underway. They're still a little bit further out. The idea of this project was proof of concept. So can we make it work? Does it work? Also, we had well-described genomes for the species we worked with. That's another problem. If you develop tools like we did that are based on gene information, you ideally have information about the genomes. But for definites, we do have that. For some algae species, we do have that. But for some of the more rare, I work a lot with endangered fishes like sturgeon, well, we don't. So how do we generate that information? So there's a couple of other things to be considered. But yeah, it's a good point. And I think there's huge need for expanding that.
Okay, great. What about, I mean, so if we think about marine organisms specifically, I personally, when I think about this, I come from an environmental chemistry perspective, and I think the chemistry of the chemical is different to start with. And then the organisms themselves, you know, if you could develop the chip with those organisms, would we take care of the differences in the organism, like a marine versus a freshwater? And then what do we do about the differences in chemistry? How do we consider that?
So the one idea is to expand the model. So right now, again, this is, I call it the core piece. Once you understand all of that, you're going to have toxicokinetics, toxicodynamics, which is different depending on what chemical type you look at in a marine or freshwater or brackish environment. It's different in different organisms. So I think there's a lot of front-end modeling on the chemistry side. So this really, before you have this molecular initiating event, the chemical has to get into or to the target site where it's causing what it's causing. This is not part of this project, but there's a lot of talk about it. How good are we to use an embryo model, for example, to simulate exposure? We have a chorion around a fish, or you have a chicken egg that's around the chicken. So how does the chemical get in there? How realistic are you? These are all questions we haven't answered yet. But that's where some of the in silico modeling comes in. So there's a whole world on, okay, our understanding of how chemicals end up where, how they get into an organism, how they eliminate it, that we try to layer onto that. But again, this is more future stories. We are aware of that, but it's something that we're not doing at this point yet.
Well, and I think, I think what you're describing is kind of the neat aspect of our field, and that we need so many different types of experts, and we need to think about so many aspects of the system. And so you're certainly focused on one part of it, but it sounds like there's plenty of people thinking about the other, how this fits into the framing of their own discipline, and so on. Okay, here is a question. Do you see the ability to use this in remote lab field situations in the future? So could somebody take this and go out into the field and use it to assess field situations?
I'm not sure about the near future. I definitely see a future for that. So if you think about the rapidness in which those tools have evolved, we now have field-based sequencers. I have a mobile laboratory. I can go up to northern Saskatchewan where there's no power, no labs, nothing, and can go out. I can extract genetic material very easily from an organism in the field now and run that on a small handheld device. It's not as powerful. It doesn't have the output, but the tools are coming out. They're really expensive right now. That's one of the biggest issues, and they haven't been validated sufficiently to give us data that we would rely on a regulatory side of things. But I foresee, give it 5 to 10 years, we will have tools that allow us to easily do that. So that's my vision and my dream, to get away from the lab scenario, but really go out into the environment and make assessments right here, right there, that are meaningful and allow us to respond in a timely manner as well.
So, okay, so that was actually one of my former grad students who asked that question, and I know she's a consultant. So could we, in some point in the future, do you see her being able to purchase this, go out in the field, take a sample, and figure out what's happening? Is that how easy this could be at some point?
Well, I think you need some expertise. So you will need to have some mini field lab, you need power for sure, or some really powerful batteries, and the instruments will probably be requiring expert handling. It's not like that you just take a drop of water, put it in a plate, shake it, and then you look at it and say, oh, we have a problem, Houston. So that I don't see that. But with a minimum lab equipment, you can probably go there. But again, this is future. We're not there yet. I just want to caution that while I'm dreaming of this, we're still a couple of years away, I would say, at least.
Well, I like that you can come here and tell us where we are, and then we can also be thinking looking forward, which, I want you to do that with us. But I recognize that you're saying we're not there yet. Yes. I have another question. Sorry, this is from a student. And I think this is actually a good question. And I personally was interested in some version of this question. And that is, do you see that there would be, do you still see a need for whole organism testing? Or do you think at some point we may be able to eliminate that completely?
Again, I think that's future. We hope we can eliminate that completely. I think that's the ultimate goal. But again, as the Holy Grail, it's hard to get to, but you should strive to do that. At this point, I think we can make pretty good predictions for whole organism tests. Again, these are whole organisms. These are embryos. So ideally, we get away from that as well. But at this time, at least they're not considered live animals. And they only require very short and rather painless scenarios, rather than having a 2-month study with an adult or an animal. But yeah, right now it's more prioritization we can do with those tools. So if we identify issues, then we can say, okay, Houston, we probably have a problem, or we haven't proven that yet. So we probably would have to still follow up with some in vivo based classic assays. But the powerful approach here right now is, if you have 100,000 chemicals you have to test, you can screen them with a very rapid, high throughput, non-animal test like the Ecotoxchip. Out of those 100,000 chemicals, probably a fraction will be problematic. So if you can identify and prioritize the ones that are problematic, and you end up with 5,000 chemicals out of 100,000 that require further testing, even if you have to run those in a whole animal test, you just cut down your testing requirements by 95%. I think that's really what we can say right now we're able to do. In the future, we may get to a point, and I hope we will get to a point where we can completely eliminate that, but I don't think we're there yet.
Okay, great. I want to conclude with one final question. We have about 3 minutes left. And there's two questions that are similar, and they're both related to communication. So you work in a field that's very complex, and I would assume that a lot of people don't start out understanding what you were talking about. And so what are barriers for you in communicating, and what are tricks that you're able to use to help you convey your science to a general public or to a regulator, so to people who don't have the background knowledge that you do?
Well, I hope I didn't disqualify myself today. But one thing I learned through this project, which was the unique feature, I talked about the GELS component, the social science component. So we had a whole team of social scientists that partnered on this project, that went out and literally talked to people. They talked to regulators, they talked to community members, they talked to industry partners. And when we started the project, when we wrote the grant, it's like, why do we need social scientists? We scientists, we know what we do. It's cool. We have all those fancy things we can, and we'd be pretty sure and confident we can. We learned so much about the lack of communication there is. Just taking the regulatory community. If you take decision makers, they're very often not scientists, or have scientists from a very different area. They speak a different language. And learning that language, I think, is critical. And I'm learning that, that older I get, the more social scientist interest person I get, and I'm getting away from the natural science, which still fascinates me. But this communication, asking questions, talking to people and listening, and you very often see this, well, I have no idea, I have no clue what you talk about. So how do I translate that? I think that was my biggest learning exercise through this project. And I think we can do a better job, and I think we can be very successful in doing that, which we have seen now. So we have like partners ranging from the OECD to just pure regulators that have no science background, that they love to play. They think that's fascinating, and that's the first time that they say, oh, I understand. I never knew that this is possible, or that this is how this all fits together.
Yeah,
well, we're not there yet. Again, there's a lot of barriers still to break down, which, well, otherwise they wouldn't need us if this would all be solved already. But it's a fascinating field. I really love to explore more.
Yeah. Well, thank you so much, Markus. I really do appreciate having you here.
I have a quick question for you. Yes. So I see there's a whole bunch of questions in the chat that we haven't answered. So if people want, they're more than welcome to send those to me. If they still like to get answers to those, I'm happy to do that. Or if you want to compile them, I'm not sure how this would work.
I thank you for that, because that's a really, I do know that people, when I'm not able to get to their questions, that's disappointing to them. So I really appreciate you making that offer. And so I would say, if you do have a question that wasn't answered, please contact Markus directly. And what I have learned is you might have to follow up. Is that okay to say, Markus?
Yell at me.
But if we can compile them, we'll also do that. Yeah. So thank you. And we have, in 2 weeks, we have our next speaker. And this is going to be, if you were able to join us for the Janie Ingram, who was able to talk about work on the Navajo Nation, we have another, one of her actually former students, who's going to talk about a grassroots project that was able to lead to a potable water supply in Sanders, Arizona. So this will be another really interesting applied discussion about how our field intersects with society. And so I want to thank everybody for coming, and we look forward to seeing you on January 28th. Thank you.
Thanks.
Thank you very much. I really appreciate that.
Yeah. Thank you, Markus.